Thank you, Melina. Hello and good morning.
Stephanie y Melina por
I want to say thank you for inviting me to talk here in this wonderful region about the advances that we have in the area of the Angelman syndrome. And unfortunately, my Spanish is not enough good for being able to talk about this. But anyway, I'll change to English during the rest of the presentation.
I'm going to talk about clinical trials, and then I'm going to do an update on the ongoing trials, which are actually very exciting. So I have a lot of disclosures I work with a lot of companies for genetic, neurodevelopmental, and neurodegenerative disorders to bring new treatments that target the underlying disorder to patients. And I'm an investigator on all three of the Roche, Ionis, and Genetics, and Ultragenyx Angelman trials. So as a result, I am investigating in the success of all of them for my patients because we want to bring treatments to patients as fast as possible. So what I'm going to do is talk about the drug development process and clinical trials for drug approval and why we have them and what we have to do in them, trial participation, outcome measures that we have to use in the trials, EEG biomarkers and natural history, and then I'm going to talk about the mechanism of the different drugs that are in trials and an update on the actual trials that are going on in Angelman syndrome. And so this is a combination of two talks on the schedule, and then Allison and I are going to come up and answer questions at the end, I think. So this is the process of drug development. It's not a short process. Why do we have to have clinical trials and drug development? Because otherwise we can't get drugs approved and we can't use them in the clinic. And then we can't make progress on having better treatments for people. So the full process of drug development is quite long. I don't think this thing is working anymore. But anyway, it involves basic research, it involves finding targets, developing assays, doing a lot of stuff to figure out what drug you're even going to test, and then doing some of that preclinical efficacy that you were just seeing, and then safety studies, and then finally you go into clinical trials, and then you have to do phase 1, 2, and then later phase trials. It can be longer than 5 years if the trials have to be long trials, and how long a trial takes depends a lot on recruitment, participant availability, how fast sites can enroll. We'd like to believe that a drug for Angelman syndrome will be approved by 2027, and we actually have a video about that. Anyone wants to look at it. Okay, so the process of clinical trials starts with phase 1, where you're doing safety. Often this is normal volunteers. Sometimes if the delivery risk is higher, like it's an IV or an intrathecal therapy, then you do phase 1 in patients. This is to rule out a major side effect and make sure the drug doesn't have some huge toxicity. In phase 2, we may combine it with phase 1 if it's a rare disease. So like, for instance, in Angelman syndrome, we've done phase 1 and 2 together, because the population is limited. And in at this phase, we're trying to figure out what dose to use. We're looking for an efficacy signal. And these may or may not be placebo-controlled, depending on the disease. In phase 3, we're looking at the efficacy of the drug. This is your registration trial that we are going to go to FDA with, and we're going to prove that the drug actually works on something meaningful to patients in the disease. In some cases, phase 2 is combined with phase 3 in ultra-rare diseases where the patient population isn't big enough. Angelman syndrome is not, that's not the kind of disease that Angelman is. So these trials are necessary for regulatory approval, and typically they are placebo-controlled. Phase 4 is a post-marketing trial. Maybe you got an approval based on a biomarker. Maybe you got, maybe information that needs to be followed after the drug is approved, and so there's a post-approval trial that gets run. So basically, the way things have looked in Angelman syndrome is there's a phase 1-2 trials. Patients go into a long-term extension because if they're on a drug, we don't necessarily want to take it away. And then patients in phase 3 will also probably go into long-term extensions. And those long-term extensions are important to get safety data so that you can take that to the FDA. You can have longer-term safety data and show that the drug is going to be safe over years. And sometimes sometimes everybody spills into the same long-term extension. But so we have a number of drugs that are on this path. GTX 102 is the Ultragenyx drug, which is it's finished phase 1-2 and it's going toward phase 3. Ionis, Ion 582 is, finished with phase 1-2 and headed for phase 3. Rugonersen is done with phase 1-2. That's the Roche drug, but it is not moving forward into a trial unless Roche gets a partner to move that forward. And then Alogabat, which is the Roche drug that works on GABA mechanisms, and NNZ-2581, which I'm going to talk about, is the Neuron drug, and those are just completing an initial phase 2. Well, the Alogabat is still in the phase 1-2 study, and the Neuron drug just completed. So we have lots of things that will move forward. But first, let's talk a little bit about clinical trials. Now, I know the clinical trials are not in Latin America yet. I think the hope is that eventually that will happen, and so when they get here, you will know, you'll have an idea what this is like and what the participation is like and everything that kind of goes into this. So we have a lot of design issues. We have to think a lot before we do a clinical trial on how to best do it. We need to think about whether the trial should be open label or placebo-controlled, and how long should the placebo period be so that we can compare the placebo to the treated patients and see a result. And what's the dosing? Is the dosing the same as in a typical person, or is it just, or is there a special dosing for people with the disease? We need preclinical studies. Sometimes we have a choice of animal models, and we have to think about that. When patients can't actually report what's wrong with them, as in Angelman syndrome, we have to think about how do we do safety assessments. We have to think about what is the age of patients we should enroll. Younger patients might respond better, but they might not. And so there's always a lot of unanswered questions in this. What's the formulation of the drug? How are we going to give it? If it's an oral drug, can we give it in a form like a liquid or a chewable or a dissolvable if patients can't swallow pills? Should we allow other medications or not? Are we looking for a pure signal on the disease versus improvement over available treatment? And what kinds of measures can we use to show that the patients are better? And this is often not well developed for neurodevelopmental disorders, because the patients that we have with neurodevelopmental disorders are not like typical patients for which most of the tests are developed. And then with a rare disease, you always have distance and travel, and patients often don't live near the study sites. So these are all things we have to think about. And what we worry about when we start trials is recruitment. Will patients be interested in being in the trial? Can we recruit them without being coercive? Are families worried about side effects? Will the families understand the difference between trial and clinical care? Will families be able to stay in the trial until the end of it and do everything? What are the best assessments? How can we do the assessments so that the participants will participate in the assessments? How can we get, really accurate reporting? And how can we maintain the blind and avoid bias in placebo-controlled trials? Some of the strategies that we use to solve problems like that are to really have an available PI and investigators that can be supportive, explain things, spend as long as needed with families and give families the big picture. And then, having an easy route to reach someone at the study site. And then an understanding on the part of families that when we are doing a clinical trial, it's an experiment. It's not clinical, it's not management in clinic. This isn't like a treatment visit. This is still, an experiment. And we're studying effects in a very controlled fashion. So it's not just like the way we do clinical medicine necessarily. We need a stringent protocol and we need rules. And it's critical to follow the rules or the study might fail to show a result that we want it to. And then in terms of retention, we try to make the unpleasant things as easy as possible, but in trials we have to do a lot of procedures, so they're going to be unpleasant things. We use motivators and rewards, try to provide families scheduled. We ask families to bring things to keep them busy, because we all know the hospital does not always run on time. And sometimes there's an emergency patient that has to go in front of your child to do something. So we try to minimize discomfort in ways that are possible. We try to optimize the time of day and the delivery of assessments with experienced examiners, give people breaks, be very consistent. We try to emphasize accuracy in terms of symptom reporting and explain to families it's very important not to embellish symptoms or answer a question one way because you think it will help you get into a trial. It's important to just tell the truth. And then, this is very important because we are going to have phase 3 trials in Angelman syndrome, and they're going to be placebo-controlled or sham-controlled. In other words, there's going to be a control group that's not getting the drug. We do not report things that might be unblinding or introduce bias on social media. We do not even go on social media and talk about our symptoms or the side effects or the positives or anything. We do not show that video of our child do something for the first time. We do not do that. Because when that happens, it tends to increase the placebo effect, and it might prevent us from getting a drug approved. We have a drug like that in Fragile X, and people are still waiting for it to be approved because of this kind of behavior. So we want to make sure that everyone knows that we do not want to do that. So being in trial is a lot of work, especially when your child requires more care than a typical child. We always tell families you have to educate yourself on what's involved and think about whether you can do it. Are you okay with infusions that might have to be delivered into the spinal fluid, or even for gene therapy into the ventricles in the brain? How long will this trial go on, and can you participate? Can you do everything? Can you do everything according to the rules? And then you have to, the diaries and the assessment forms are just endless and they seem like a big pain. Can you do those accurately, honestly, and get them all done? Because if you can't, probably someone else who can help us more with getting the drug approved should be in the trial. And that seems harsh, but it's a truth. If participants pull out or don't do their activities, then we might need to recruit people to fill the space. We may have incomplete data. It may delay the trial finish. And getting to completion is really the goal. We want to minimize people who will leave the trial because it's too stressful. We want the people that join to be all in, and they are going to do everything. Because others are depending on people in trials for their future treatment, and you don't want to let them down. And you're in partnership with your trial team and your study sponsor. Safety in trials is really important. We have to report anything that happens to you, even if it's not due to the drug. We have to report all recent procedures. We have to report any new medications, even if it's just some herbal thing or a vitamin that you got at the store. So we really have to keep track of everything. And you can't be selective about what side effects to report. We have to report everything, and we just say it's not due to the drug if it's not. There's often issues with travel. We've learned a lot about how to do more things at home during COVID. So we have some trials where we have a lot of video conferencing visits, or we have outcome measures that are done in the home. We always need more sites. We need trial networks. Travel is usually paid by the sponsor and industry-sponsored trials, but there may be budget limits, especially in federally funded trials. So these are all the things that face us when we're thinking about a trial with Angelman syndrome. And we have to work together as partners. So these are just me with some of my trial participants in all different diseases, and we really get to know the families and work together as partners to make sure everything gets done right. Okay, now I'm going to transition to talk about outcome measures a little bit. This is a big hurdle for kids with neurodevelopmental disorders, including Angelman syndrome. You often have to test a broad functional range. You have patients who are off scale for the measures that would be appropriate for their age. And so we have to find measures that we can use in a special population that wasn't validated in a special population. And then we need a lot of information ahead of time to see how it works. And then it's always not clear how well these measures that were created for typical people get at the problems that a patient with a developmental disorder like Angelman has. Sometimes there's a real problem with cooperativity of the patient actually doing the measure when we want them to. And then we have this issue where the FDA wants us to pick one thing that will tell us that, yes, that trial was successful, when the disease has hundreds of problems associated with it. So that's another difficulty, is trying to even convince the FDA that composite or MDRI, measures that measure a big part of the disease, will be helpful. So we often need to design special measures for the disease before the trial. So all this has been a challenge, but a lot of it has been overcome in designing the current Angelman trials, much thanks to the work of FAST that Allison talked about. And so we have 4 basic kinds of outcome measures in a trial. They're performance-based measures where the person has to perform. This would be like a cognitive test. There are patient-reported outcomes where the patient reports their own symptoms. This obviously does not happen in Angelman syndrome. Observer-recorded outcomes where the caregiver reports what the patient is doing, and then clinician-reported outcomes where the clinician reports what the patient is doing. And these have to be acceptable to the FDA to run the trial. If the FDA doesn't say ahead of time this will be a good enough measure for approval, then you may not get approval. So we have to make sure that that is okay. In Angelman syndrome, we've picked measures that measure things that came out. Allison talked about the Angelman syndrome disease concept model, and this is where you identify the most important areas to measure based on what families and clinicians think are the biggest problems. And so in Angelman syndrome, communication was the highest, the most endorsed problem, and so we measure communication, but also cognitive and adaptive functioning, fine motor development, and gross motor development, which were all flagged as problems. So we use the Bayley scales of infant development. Angelman families that have to watch their child do the Bayley do not love the Bayley, but the Bayley is kind of the best thing we have for development in Angelman syndrome, even though it's not ideal. It assesses skills in different domains: cognitive, language, motor. We have a lot of data on it historically. It basically has a age range of function of 0 to about 3.5 years. And most Angelman patients, virtually all Angelman patients, will fall in that mental age. And so therefore it's addressing things that are within. I'm good, I got water. It's just a chronic throat clearing. So it's a performance-based assessment. They have to sit with an examiner and they have to do certain things that allows us to assess where their development is at the time that they're being tested. Now, obviously, sometimes they don't do what they're supposed to do, and then that makes it difficult to assess their level of function. This is where the trained clinician comes in. This is used in many studies in Angelman in clinical settings. It's used in other diseases, and it's really providing a basis for comparison. If we have natural history data, which I'll show you on the Bayley scale, then we can compare the trial data to the natural history data. So it is sensitive to change over time, not over like a month, but over like a year. And it's important that the Bayley be administered when the individual is most likely to cooperate, like if they're a morning person in the morning, and by the same clinician as much as possible, and by using motivating things, which often are treats or snacks, but could be something else. The Vineland is an adaptive behavior scale. It's an interview with the family about how does this person function in life in different domains, like communication, activities of daily living, socialization, motor skills. It's an observer-reported measure. A clinician interviews the caregiver who knows the patient well, and the caregiver reports on what the patient can and can't do. And it's administered in a very standardized way. For this, it's really important to ensure that there's enough time. Sometimes when the family is there with their Angelman child in clinic and they're running around after their child, it's not a good time to do the Vineland. So you have to schedule it at a time when the caregiver can really pay attention. The caregiver needs to the same caregiver needs to complete the interview, and we have to administer it with the same clinician as much as possible. Now, this is another key measure that we are using in trials. And then there's something called the Observer Reported Communication Ability Scale, or the ORCA. And this was something that FAST worked on developing. The FDA was involved in the development of this. The ORCA is based on the premise that communication in Angelman syndrome is very behind, and there's a lot of impairment there. So an improvement in communication that might be very meaningful to families and might be very helpful for an Angelman patient might not be picked up on a regular scale for typical child communication development. And so this is a more granular approach to looking at communication. It was based on interviews with Angelman family about what's important in communication, and so we are using that in the trials also. And so we also, have to just make sure that the caregiver is completing the questionnaire and that the same person is completing it. Then there's, I'm just going to put all these on. There are CGI scales. These clinical global impression this is designed to be a measure like what your doctor would do in clinic with you. If your doctor starts you on a new seizure medicine, when you come back, they're going to ask, are the seizures better? How long did they last? How many did they have? We're essentially assessing how is your child doing in all these domains, and trying to understand whether there's been improvement or worsening, or whether things are the same. And so we measure with the CGIs that we've developed for Angelman syndrome. We again designed them and chose the domains based on that disease concept model. So we're looking at sleep, behavior, communication skills, gross motor skills, fine motor and oral motor, meaning like drinking and eating skills, and activities of daily living. In the Ultragenics CGI, then there's one that was created by Roche, which has similar domains to the Ultragenics, but also covers seizures and is not anchored with specific, like, this is how you should rate a 3 and a, oops, whoa, all right, now, can I go back? Yeah, okay, this thing, all right, my clicker had a seizure. Okay, so, it's not as anchored, it's more kind of free form, so there's some differences between these two, but they're both being used right now. This is natural history. This is very important. The people who have worked tirelessly for decades to collect this natural history are very critical to the treatment development effort for Angelman syndrome. Because if you know what Angelman patients did over time in the past, then you can compare what are they doing when they're on an intervention or a drug. And as we can see from this, if you look at the red, the developmental trajectory is pretty flat. The gray is normal development, and it only goes up to 3.5 because that's as high as the Bailey scales go. But the red is Angelman development over, out up to almost age 16. The blue is non-deletion, and the red is deletion Angelman syndrome. So you can see there's not a lot of development. There's some development that goes on, but there's not a lot. And we need to know how much that is so that we can compare it in trials. And you'll see some plots where we're using that natural history. The EEG is another thing that's been very helpful in Angelman syndrome. If we're seeing a clinical improvement, or maybe even before we see a clinical improvement, we would love to see that we're changing the brain in an objective way where we don't have to rely on whether the Angelman patient was going to cooperate with the Bailey that day. So the EEG is a way that we can look at that. So we put the leads on and we measure the brain waves, and Angelman patients have an increase in those squiggly lines right over here that are called delta power. They're discharges that are moving or moving at a certain frequency in the brain. And when we correlate the amount of delta power, Angelman patients have too much delta power. When we correlate the amount of delta power with cognition, we can see the more delta power they have, the lower their cognition is. And in fact, if we try to predict what their cognitive score would be from the delta power and compare that to their actual cognitive score, we get a very nice line right here that shows that EEG is really giving us a tight idea of cognition in Angelman syndrome. So we have a good biomarker, and that's been employed in trials also. All right, so now I'm going to shift over to talking about the actual results of trials that have been run. So Neuron is running a study of their drug called NNZ-2591. And by way of a little background, this drug is very similar to trofinetide, which is, they're both synthetic analogs of cyclic glycine proline, which is a part of growth hormone, or IGF-1, which is a peptide that occurs naturally in the brain and has had positive studies. So the trofinetide had positive studies in Rett and Fragile X syndrome. It's now FDA approved for Rett based on improvement in that trial. The NNZ-2591 is not trofinetide. It's a more stable, better, easier to get through the stomach, less side effects molecule that crosses the blood-brain barrier better than trofinetide. So it's a better drug. And now, how it exactly works, I think we would be deluding ourselves if we said we know, but it mimics some of the natural actions of this cyclic glycine proline, which are reducing overactivity in neural signaling pathways to promote synapse or brain connection formation, like others have talked about, rescuing abnormal shape of the dendrites on neurons, so changing the structure of the brain connections to be more normal, and stimulating protein synthesis at the synapses to kind of help with the synaptic health. And the effects of this drug are state dependent. In other words, they correct impairments or problems with the neurons, but they don't affect what's going on normally. In the Angelman syndrome mouse, NNZ-2591 improved impairments in locomotor activity or the hypoactivity in the mouse, motor performance, spatial memory, learning, social interaction, and seizures. So it looked promising. So Neuron went forward with a study of NNZ in a phase 2 trial in children with Angelman syndrome in Australia. And this was a small phase 2 study. It was open label. In other words, no one got placebo. And so patients had a screening period where they came in and we looked at their characteristics, made sure they qualified, and then they went into the treatment period. There was a dose adjustment period at the beginning, and then they stayed on the top dose. In fact, I think all the patients stayed on the top dose. And then they had measurements, some of those measurements that I've just talked about, but mostly the clinical global impression scales, and then they had a caregiver global impression of how the patient was doing that studied kind of the same domains as the clinicians rated. And they did the Bailey, but this study was only 3 months, so that's kind of too short a time frame for the Bailey to really show change. So this is the results that the Neuron has presented from the trial. The CGI showed an overall improvement. The line here is no change. Most of the patients are above the line, so they're showing improvement based on the clinician rating. Here, this line would be no change, and over here would be worse, over here would be better, or over here is better, over here is worse. And so the patients were improved in basically all of the domains that were measured on the CGI. Again, it's open label, so some of this could be placebo, and probably is placebo, but hopefully not all of it. And then this is the Angelman, this is the clinician, this is the caregiver rating form where there was improvement in many of the patients, and again, improvement in the domains. And then they looked at just the children aged 3 to 12, and all of those children improved. And this is the idea that younger children might have a better response sometimes to these treatments. So this looked very positive. Some of the things that were reported in the trial are quoted here. Patients were sleeping a little bit better. They were calmer, more focused, seemed to understand more, sometimes were steadier on their feet. And these same kinds of things were reported by both clinicians and caregivers, although some of what the clinicians report comes from the caregivers. So there's just, but in general, there was a sense that something was happening. And so this will move on to a phase 3 trial, which will be placebo-controlled. So we will then know how much is placebo. Okay, I'm going to talk about the antisense oligonucleotide trials. And so you've seen similar renditions of this drawing. Again, this is mom's gene, where this is the maternal gene, where all of these genes here are shut down, including the UBE3A antisense gene, and so UBE3A is made. This is a normal person. Here, the UBE3A on the dad's gene is active, and so UBE3A is shut down on dad's gene. In Angelman syndrome, mom's gene is gone, dad's gene is still shut down. We have an ASO that can go in there and work on this transcript, the antisense transcript, and kind of knock it out, and now dad's gene can be transcribed and made into protein. So this is basically the same thing that multiple other people have told you. And when we do that in the mouse, the Angelman syndrome, the UBE3A RNA goes up, the UBE3A protein goes up, and the antisense RNA goes down, and so that's good, and we can see the protein being made on a blot, and we can see the protein being made in the brain. And so in the mouse model, this seemed to work. And so we went into trials in Angelman syndrome. And these are all phase 1-2 trial. They're all first in human, multiple ascending dose trials. Means you start at a low dose, make sure the patient is safe, and then move to a high dose. Roche Genetics originally, and then after Ultragenyx brought their drug, Ultragenyx, and Ionis have all been in these trials. And so Roche and Genetics escalated the dose within patient. Ionis had a dose escalation in cohorts. All had very similar safety objectives. So they're looking at safety, they're looking at pharmacokinetics, they're looking at clinical potential efficacy in multiple functional domains of Angelman syndrome that again come from the disease concept model. So communication with the ORCA and the Vineland and the Bailey, sleep with the diarrhea and EEGs and this CGI, behavior with a behavior scale called the ABC and the CGI, gross motor with genetics had the Actimyo, which was a sensor that you could wear and look how well you were walking. Vineland and Bailey also, fine motor, Vineland and Bailey, seizures, diarrhea, EEGs, CGIs, the EEG biomarker, and then standardized videos, which Ionis has used, which are really kind of nice to try to look at daily things that Angelman patients are doing on video. So Roche, so I have 29 seconds left up here, but I'm going right into the second talk, okay? Okay. So Roche had the rugonersen trial called Tangelo, and they had doses, different cohorts that had ascending dose within the cohort, but the dose also ascended between cohorts. So these cohorts were progressively getting higher doses, and then there was a long break, and then there was a bridging dose, and then there was a long break, and then there was a long-term extension. Then these breaks had to do with getting adequate safety data to dose the patients more. And so they had 61 patients, age 1 to 12. Mutations and deletions were included. The patients were underdosed. They got dosed every 4 to 6 months, and 6 months was definitely too long to go. Patients were coming off the drug at the end of and we could tell that because their behavior and their sleep got worse. Side effects were limited going to the maximum dose that they wanted to go to. And they had some side effects of post-dose fevers and ataxia, sometimes vomiting. These could easily be managed and were not really a limiting problem. And what did Roche see in their study? In a nutshell, I'm going to summarize these studies really quickly because of time. But what Roche saw was that on that EEG biomarker, remember the delta power drifts down with age. It gets a little better as you get older with Angelman syndrome, but in the trial, it dropped way down in the multiple ascending dose part, and then it stayed low on the patients that were getting dosed every 4 weeks. You know, we have to remember we're doing the EEG when the patient comes back in there for their infusion, so we're at a trough level where the patient's been 4 or 6 months since their infusion, and so in the 6-month patients, the EEG biomarker did not show an effect, showing that really that was too long a time to go between dosing. So, but this is very nice to be able to see that you're changing the brain and it's becoming more normal. And then they also had likewise clinical data where they compared the Bailey, the amount of improvement on the Bailey during the multiple ascending dose and the long-term extension part to natural history. And so this is the change relative to natural history. So if you took the amount of change a patient with Angelman has in natural history for a year, and then take the amount of change they had for a year in the trial, and subtract the normal change from what you had in the trial, this is what you have left over. is this amount of change. And so patients were changing more. They were making more improvement on the Bailey, really across all the domains, except for gross motor didn't show much change. And that's not that they weren't changing, it's the way the Bailey measures gross motor. But across all of the communication domains, cognition and fine motor, they were seeing a difference between the patients on treatment and natural history. Okay, so that's what I'm going to say about that, because again, I don't have a lot of time with each of these studies. So the Ionis-Halos trial of Ion 582, so they also had these different cohorts with different doses, started with a really low dose and then went to a medium dose, and then there was a high dose, and then depending on what you went into at the beginning, we now have patients who had continued dosing and are in a long-term extension either on the medium dose or on the high dose. And so these were also mutation and deletion patients, and there was a broad age range. There were the pink patients were 4 to 17, the green patients were 2 to 5, and cohort E was older patients, 18 to 45. This was the Roche, in this trial we've seen generally good tolerability. Most of the adverse events have been typical things that kids got, like vomiting illnesses or upper respiratory infections or fevers, and there weren't any kinds of other side effects specifically related to the ASO. And nobody left the trial because of a side effect due to the ASO. Okay, so what have we seen from that? So this is data that Ionis just presented at the Angelman Syndrome Foundation. And again, we see this is natural history, this is the patients on drug, and they're making more progress in these measures we call GSVs, which are growth scale values that show how much growth you've had on the measure. And then also on the Vineland, they made more progress than natural history, and that's in receptive communication. In expressive communication, we see the same thing. More progress than natural history. More progress than natural history on both the Bailey and the Vineland. They're corresponding to each other. And then in the CGI, the doctors who are rating the patients are saying that they're getting better, they're improving on the CGI. In that case, improvement is negative. Okay, so in communication on the ORCA, these patients made more improvement than natural history, and they made more improvement than what would be statistically considered clinically meaningful based on studies that were done with the ORCA, which is more than 2 points. So again, improvement. Cognition on the Bailey shows an improvement over natural history and shows an improvement in the clinician's ratings of the patient. So across everything, we're seeing improvement, and this is just all of these measures put on one graph, and you can see that for most of the patients, for a high percentage of the patients, over 50% of the patients, there's improvement in almost all of these measures, except for the gross motor domain in the Bailey. So there's quite a bit of variability between patients, obviously. Like some patients are improving more in one thing and less in another thing, but the global picture is one of improvement over natural history. And then these are just interviews that Ionis did with families that show that families were reporting things like the patients can focus on what the caregiver tells them to do. They can communicate their needs, wants, and thoughts, and they can move around their environment better. They can sleep better. They can take care of their daily needs better. And these are, most families were reporting that these were a lot better. Again, there's variability, and there's going to be that in all clinical trials, but in general, improvement. So the HALOS study did show consistent benefit across treatment in people with Angelman syndrome, favorable safety and tolerability at all dose levels. HALOS also has reductions in EEG delta power observed at 6 months. I didn't show that. There's evidence of clinical improvement across all of these domains, consistent benefit across all ages, and there's not really been an age effect seen yet in any of these trials. So the conclusion that we've got a great phase 2 study, we need to go forward in a controlled study in phase 3 and really demonstrate that the drug works to the FDA. Okay, genetics and ultragenics. They called their trial Kick-Ass. I have to think that was invented by Allison or somebody. So they had the same thing where you have different doses that you give back to back, and they had a problem at the beginning of the trial, which unfortunately occurred at our site. The first 5 patients treated with a 20 to 36 milligram dose all got a radiculopathy, where the roots coming out of the spinal cord got involved, and then the patients got weak in their legs. And these patients did recover, and ultimately the way of getting around this was to lower the dose, use a flush, in other words, put in more fluid after you give the dose, put the patients in Trendelenburg with their head down, and give steroids before the loading doses. And so this strategy has actually mitigated. There have been some suggestions of mild problems with this since they went to the lower dose strategy, but in general, this has prevented the problem from happening. And so they've treated 83 patients stratified by age in different cohorts of this study. And when they look at their data, they see kind of a similar thing, and they haven't had any other really big safety issues. It was really just avoiding that one problem. So on the Bailey 4, they're plotting out a cohort that was started more recently, so it doesn't have as long-term data in blue, and this is a purple cohort that's been going on for a long time. This one's only 15 patients, but they're showing the same trajectory of improvement in cognition relative to here we have natural history plotted over here, so the same kind of thing as we've seen in the other studies. Here's the receptive communication improving, way more than natural history. And then this is on the CGI. You can see the sleep improving. Down is better here. And you can see the behavior improving. So they're looking at sleep and behavior here. And then this is just another way of looking at that as a responder analysis. So sleep and behavior both seem to be getting better. This is another one of those plots that's like a heat map where you show who's responded to what. And as you can see, there's a lot of variability across patients, but there are a lot of responses. And this is called a multi-domain responder index, where you get +1 if you respond in a statistically significant, meaningful way. In other words, you have to get over, like, for instance, 6 points in receptive communication and 5 points in cognition. And then for each domain you improve in, you get 1 point. And so most of the patients here are positive across the board for both studies. So there's a global improvement that we can see. And this is just one way of looking at multiple symptoms at the same time. And then overall, they see an improvement in the CGI. And then this is their EEG data, which shows that delta power is coming down as well. And then they also looked at sleep spindles, which showed that sleep spindles were increasing, which is good because Angelman patients have trouble with sleep spindles, and they also have trouble with sleep. And so this is an EEG signature of improved sleep. And then further data is that those 5 patients that had to come off of their drug, and then they had to be off for like 2 years, they've now, 3 of them at the time of making the slide have gone back on the drug and been on it enough time, where we can see that all of these green areas where they made gains, they lost it during those 2 years, and then when they went back on the ASO, they gained it. So that's like an on, off, on kind of methodology, and really suggests that those effects were due to the drug and not just due to development. So now I'm going to show you a couple of videos. Patient 1 is in one of the trials, and I'm hoping this is going to trigger the video, but I don't know if it's going. You guys know how the video is triggered? Oh, here we go. Okay. So she's in the middle, and she's sitting on the trampoline here. This is just an example of the kind of thing we're seeing. And it's kind of the human equivalent of Allison's mice that she showed you on the rotarod. So she's sitting on the trampoline. She likes to be with her sisters. She wants to be on the trampoline. She's actually reasonably coordinated for Angelman syndrome, and she can get up and walk around a little bit on it, but she really can't jump on it. This is her after 6 months after 5 doses of an ASO, so about 6 months. And I'm going to try it. And that's her now. She can jump on the trampoline, so she's like the mouse. She's not falling off the rotarod. She can go down to her butt and jump back up onto her feet. So this is, when the family sent us this video, we were like, wow. And they've consented to have the video on the screen. Okay, so that. Okay, and now I'm just going to show one more video. This is a patient who is, she was just kind of thinking about learning to walk, but not quite walking yet when she came into the study. And here she is. She's on her tiptoes, very much on her tiptoes. So much on her tiptoes that she really can't balance, and she kind of falls over. And then, just to speed things up here, I'm going to start the next video. So then here she is. She's at she's at an Angelman syndrome walk, and she's going to come around the corner, and here she is walking. She's just walked like almost a mile, and she walks through there, and she's also been on treatment about 6 months. So these are the equivalent of those mouse things. Okay, so this is all very positive, but we have a lot of issues to get to the best genetic correction to improve these phenotypes. We have a lot of things we still need to understand. What age is clinical correction possible? How much correction? Are we just going to see if we start, and how long is that going to go on? Like if we fix things and now the brain can't learn as fast as it can when you're a baby, are you just going to keep seeing improvement for decades? Or are we going to see all of the improvement up front? We don't know that. What are the time frames? Is the EEG biomarker going to predict ongoing clinical change? Will it predict clinical change even if we didn't measure cognition? What's the best dosing frequency for the ASO to make sure that patients are always on drug and not coming off drug at the end of their dosing period? How do we push brain rewiring? Should we be doing learning paradigms? Should we be doing therapy intensives? What should we be doing to improve learning? And one of the big critical agendas is how to measure the entire disease rather than having the FDA force us into one very specific outcome that the whole trial will depend on. The future, Allison talked a bit about the future beyond ASOs, is some of the things that will be one and done, where you won't have to keep going and getting injections, would be the AAV gene therapy from UPenn, encoded, has a microRNA that activates DAD's gene. We're working on the CRISPR project that Allison talked about, our big grant that activates DAD's gene. And then we're working on a stem cell mediated gene therapy to replace mom's gene. The gene therapy also replaces mom's gene. So this is the future, and I have a picture of the future there. That's what it's going to look like. I want to just acknowledge all of our team, our Angelman team at Rush, over these past 4 or 5 years when we've been doing these trials, as well as in our MRI team and our infusion pharmacy and all the people we work with, as well as our funding from foundations, FAST and the Angelman Syndrome Foundation, and the FDA that supported the natural history grant before, and then our industry partners, and then all of our Angelman syndrome families who are, as I said, our partners in making this happen. We can't do it without you. You can't do it without us. We have to work together. Thank you.